So these are my disclosures. I am an employee of Spark Therapeutics. However, today I will represent Geneton in presenting this work that we've done over the past 6 years on developing a gene therapy for Crigler-Najjar. Okay, see if I have a pointer. It's a big pointer. Okay, so first of all, of course, we're talking about gene therapy, in vivo gene therapy targeting the liver, and the liver has been for a long time an ideal target tissue for the treatment of inherited disorders for a number of reasons, which are listed here. The fact that it is a very metabolically active organ, is highly vascularized. We have a lot of experience with liver gene transfer now, mainly with hemophilia rather than other metabolic disorders. And we know that if we target the hepatocytes with a vector expressing a transgene, we induce tolerance, immunological tolerance. And so there's a number of diseases that now are explored for gene therapy, among which probably the ones that have primarily a liver involvement and no parenchymal damage are the most interesting. And so among them, Crigler-Najjar syndrome is the disease I'm going to talk about today. And it's a very attractive disease for the standpoint of gene therapy. I'll come back to that in a slide. This is, however, a very rare disease. It's less than 1 in 1 million at birth. The incidence is an autosomal recessive disorder that is caused by mutation in the UGT1A1 gene. And the result of that is that children affected by Crigler-Najjar syndrome develop a high level of unconjugated bilirubin, which can be lethal because it accumulates in the brain and leads to neurotoxicity. Today, the only curative treatment for the disease is liver transplantation. However, there is a symptomatic treatment for Crigler-Najjar syndrome, which consists of phototherapy. It's quite cumbersome because it requires 12 to 14 hours per day of phototherapy to basically catalyze the photoisomerization of bilirubin and the excretion from the kidneys. And here you can see how there are several issues related to doing phototherapy for so many hours per day. You can imagine it's definitely something that you don't want to ideally have to do. And so Crigler-Najjar remains today an important unmet medical need. As I mentioned earlier, Crigler-Najjar is an ideal model for gene therapy because the liver parenchyma is overall quite healthy. There's proof of concept studies in animal models showing that if you express the UGT1A1 transgene in the hepatocytes, you can correct the disease. The threshold for therapeutic efficacy is quite low, as in fact it is sufficient to correct 5% of the, or restore 5% of the enzyme activity, and with that you will convert the disease from severe to mild, perhaps avoiding the requirement for phototherapy. There's quite standard measures of efficacy. One of them is the measurement of bilirubin, which I'm sure all of us have done at least once in life. And then finally, which is also important, is that gene therapy, if not successful, it will not prevent liver transplantation or continuation of phototherapy. Finally, as I will show you, there's animal models that we can use to test any new therapeutic approach to the disease. Here is the standard approach. I will not use the pointer because I think it's a little disruptive. For this project, we use an AAV8 vector packaging a single-stranded genome encoding for the codon-optimized version of the UGT1A1 transgene, the human UGT1A1 transgene. This was under the transcriptional control of a HET promoter, a liver-specific promoter that has been used previously in the clinic. So we went on, and at the beginning of this project, we tested our vector first in the UGT1A1 mouse. This is a mouse model that was developed by Giulia Bortolussi and Andres Muro at the ICGB in Italy. And it's a knock-in mouse that shows basically the symptoms that are quite equivalent to those found in humans, so elevated levels of bilirubin, unconjugated bilirubin, and neurotoxicity. And in fact, to keep the animals alive, you need to expose them to phototherapy, which in a mouse that is quite hairy is kind of difficult. So what we did, we tested different doses of viral vector in this mouse model, and we basically established, as you can appreciate from the red line in the graph, okay. Anyway, from the graph, if you follow the red line, that at every vector dose, starting from 1 × 10^11 vector genome per kilogram, we observe a complete reduction down to wild-type levels of total bilirubin in serum of these animals for the duration of the study, which was in this case 9 months, and of course there was rescue of lethality. We went on also and test, and I'll come back to that in a second, the same vector construct also in the gun rat, which is, I call it here, an intermediate size model of Crigler-Najjar syndrome. This is a naturally occurring animal model of the disease. They carry the same mutation of the mouse, or rather the mouse was designed with the same mutation as the rat. And also the rat has high levels of bilirubin. And if you see the red arrow in this slide, it's very easy. You don't need genotyping to select Crigler-Najjar rats because they appear since birth with jaundice. So, however, one good thing about this animal is that they don't die out of the disease despite the high level of bilirubin, and so they don't require phototherapy, which makes things a little easier. Also, in this case, we did a dose escalation study where we tested our vector, and we showed that we did see correction, at least partial correction of total bilirubin level in the serum, starting from 5 × 10^11 vector genome per kilogram, with complete correction of the bilirubin at the dose a little higher, 5 × 10^12. What is interesting is also in these animals, because of the size, we were able to collect the bile, and so in the graph that you find on your right, you see how in the bile we were able to detect conjugated bilirubin, which is really evidence that we restored the enzymatic activity of the UGT1A1 in hepatocytes. And at the same time, the more conjugated bilirubin we found in bile, the less total bilirubin we could find in the serum. So there was a biochemical evidence that the therapy was working in the rat. However, if you follow me, you notice that the dose that we use in the mouse was quite different from that that was therapeutic in the rat. And so we wanted to explore this because, of course, when we wanted to proceed with our dose-finding studies, the question was which one is the animal that best represents what we're going to see later in humans. So this is, for example, in this case, we use a secretable transgene, human factor 9, showing that in the rat you get more or less a log lower expression than in a mouse, and also you find less genome copy number. So again, which model is the best to move forward with those finding studies? So we did an experiment. We basically monitor our rats injected with secretable factor 9, with human factor 9 transgene, and we observed that at a dose of 5e12 Vg per kilo, we were getting about 10% of normal factor 9, which interestingly is quite close to what was observed in human studies, where a dose of 2e12 vector genome per kilogram was in the range of 5 to 7%. And so essentially this study was suggesting that the rat is a more predictive animal in terms of efficiency of liver transduction that you would observe in humans. So this is what we use then for our dose-finding studies, showing that in the upper graph, doses of vector above 1 × 10^12 Vg per kilogram resulted in correction of bilirubin, and in the lower graph, we basically plotted a similar readout, where we show that in the x-axis, you basically have the vector genome copies per cell. And so above 0.6 genome copies per cell, you do observe a complete correction of total bilirubin in serum. So this slide is just to show you that we also use the rats of the, to perform our GLP tox by distribution and shedding study, which was an eventful, so the vector was well tolerated with no pathology finding, with a follow-up of 180 days in the longest follow-up arm. We also evaluated the risk of germline transmission using in situ hybridization in the rat gonads. And then here below on the left side, you will see the shedding where we don't see any vector. In this case, I believe is in the urine within a month post vector infusion. And then the right side is basically showing that the vector had the expected tropism for AV8, where the highest targeted organ was the liver. One unexpected finding, somewhat unexpected, was that the human UGT1A1 transgene in rats was immunogenic. So in fact, in all the animals that received both the low and the high dose cohort, which basically you have to follow the red box in this slide, and basically starting from day 29, we did see development of IgG against the human UGT1A1 transgene. This did not affect the expression of the transgene, however, was a potential safety concern. So to address that, what we did was to test whether this was a simply a species-specific reaction. And so what we did was to inject animals with a rat version of the UGT1A1 transgene, which on the left side, you can see how both the rat and the human transgene, they could correct the bilirubin, so they resulted in correction of the bilirubin in serum. However, believe me, this may be a little confusing without a pointer, but I don't think I can easily, okay, maybe if you can follow me. The yellow band of the positive control will tell you that the animals did not develop antibodies to the rat transgene, while they did develop antibodies to the human transgene. So I think this is important when you think about your preclinical studies, that sometimes you may see immune responses that are simply driven by the fact that we're testing oftentimes human transgenes in non-human, so in animals. This is a brief timeline. The only thing that I want to point out about that is that among all the work that was done, which I will not show you here this part, is the development of a scalable process to manufacture the vector, which then we use to both make the vector for the GLP tox studies and also the GMP vector for the clinical trial. We also did open a prescreening study, which now is closed, to identify potential participants in the trial. This was very, very useful and was something that went very well with the help of the patient organizations, because in this way we could evaluate whether we had enough patients to conduct our trial. So this study, this prescreening study, was opened in 5 clinical centers in Europe, and in about a year we identified about 50 Crigler-Najjar subjects. Now as I say, the study is closed, and actually we started a Crigler-Najjar World Registry, which has a slightly different aim. In this case, is to collect natural history of the disease. In fact, this study is open worldwide, and so far we have a clinical history from we're going towards the 200 Crigler-Najjar patients included. And just to give you a snapshot, this is work done mostly by Sam Aronson at the Academic Medical Center in Amsterdam. You could see how, with the data we collected, we could plot, for example, total bilirubin levels, and that basically shows you how there's quite a portion of the patient population where bilirubin is not well controlled. And of course, if you think about the far end on the right of the graph, those patients are unlikely to be healthy at this point because those are highly toxic level of bilirubin. So, which makes the case that perhaps having better treatment options for this patient population is important. So the clinical trial. The clinical trial is an open-label, multi-center clinical trial. It is basically a center involved are 5 clinical centers across Europe, the same centers where we run the prescreening study. And it has a dose escalation design starting at a dose with therapeutic potential. If you recall a few slides ago, I showed you that we extrapolated from the rat studies that a dose of vector above 1 × 10⁻¹² was expected to be in the therapeutic range. I'll come back to the second and third cohort in the next slide. This is, of course, is a study that involves both males and females participants, and also pediatric subjects. Is a single intervention, and then of course there's a number of parameters that are monitored, including total bilirubin, bilirubin albumin ratio, phototherapy, time in phototherapy, and so on. Here is the design. As I mentioned, there's 3 cohorts. There's a baseline period, and the reason why we have a baseline observation period is that is actually shown in this graph in the slide, where these are the bilirubin levels over time in Crigler-Najjar patients, all carrying the same exact mutation. And so you quickly realize how there's a quite high variability, which depends probably on modifier genes, but also how much time the patient will like to be in phototherapy, how so the intensity of the treatment depending oftentimes on whether the patient likes or not, can stand or not to see him or herself quite yellow, so because of the jaundice. So there's this baseline period followed by the investigational drug administration, and basically this is again about the design. In the first 2 cohorts where we actually test increasing doses of vector, only adults are included, and then in the 3rd cohort, which is an extension cohort, this is a mixed population of participants with both adults and pediatric subjects. Starting the lowest age of inclusion is 10 years of age. Here is a snapshot of the safety of the trial and where we are today. So 2 participants received the vector. The whole administration procedure, the whole trial so far has an excellent safety profile. We had no serious adverse events associated with vector administration. The infusion procedure was uneventful, and both participants are doing well. And then, as I mentioned, we have a baseline observation period, and we have additional participants lined up in this observation period, and they're ready to go for the next, for inclusion in the, for administration of the investigational drug. This is just a summary, but I want to use this slide to really think about what is the future of this therapy. So of course, or any actually gene therapy targeting hepatocytes. In animal models, we showed that liver gene transfer resulted in a safe and long-lasting and effective reduction of total bilirubin. And so, of course, now the clinical trial is helping us address to what extent this is translatable to humans, and then whether we can still rely on the fact that with AAV, of course, we transduce a subset of the hepatocytes, and whether this is sufficient to translate into therapeutic benefit. Of course, the other point, which is very important and is not only important for Crigler-Najjar, is what is the persistence of gene transfer in pediatric, in younger individuals. I'll come back to why we choose 10 years in a second, 10 years of age as a lower limit of inclusion as far as age. And so when we look at the pediatric patients and we think about now, in this case, mice, you see how the liver grows as the mouse ages, but there's quite a steep a period of steep growth early on in the life of a mouse, pretty much in the first part of the curve, and then the size of the liver is stabilized. What we learn is that if you do gene transfer in this part of the at this age of the mouse, so when the liver is growing quite fast, what you observe is what was published by Andres Muro and Giulia Bortolussi in 2014, and other groups also show the same phenomenon, which is basically you have vector genome dilution. So the proliferation of hepatocytes results in dilution of the vector genomes and partial or total loss of efficacy. So the question now is, so this is what happened in a mouse, and then what happens in a human, where, again, in this case, the graph on the right is actually the curve of growth of the human liver. And, of course, if you go very early on, you have a long way to go before you reach the adult size of the liver. And then we thought that the 10 years of age was close enough to the size of the adult liver to allow at least only for partial dilution of the vector genome, if any, let's say. But this will be tested in the clinical trial in the third cohort. Now, this is just a slide that comes from Andres Muro and Giulia Bortolussi, showing you a little bit what happened if you, this is work ongoing, if you treat the animals at 4 days of life, 11 days of life, so it will be P4, P11, or 18 days of life. And you can see how, of course, you can still, at P4, essentially you get complete dilution no matter what dose you give to the mouse, even if you give very high vector doses. These are Vg per kilo in a mouse, which tends to have a very high transduction of the liver. And then from P18, seems like you get the best results. And then this work, as I say, is ongoing to really try to draw a parallel between what you can expect in humans based on developmental stage of the liver. So, of course, that brings to the rationale for vector administration, and then I will close with that. Of course, in adults, even though we have evidence of long-lasting expression of a transgene in the liver, we may expect, for example, that we need to re-administer the vector to adjust to achieve full therapeutic effect. In young pediatric subjects, I think you got the message. We may need to re-administer the vector because you may have partial or total loss of transgene expression over time. The challenge, of course, I think anyone working with AAV knows that by now, is that when you give AAV, they're also very powerful vaccines, so you tend to vaccinate animals, they develop antibodies, and these antibodies tend to persist over time. So really you have to find ways to get around the antibody issue. Now, this is a list that is completely, I mean, it's incomplete. It's completely incomplete. And possible strategies to get around the issue of antibodies. In the lab, we work a lot on this topic, both on the side of immunomodulation, and I'll show you one slide about a possible way to do plasmapheresis. Immunomodulation, this is just a slide from a paper that we published recently showing that basically you could try to de-immunize the AAV by, for example, targeting the innate immunity to the vector, and that would result, we show that can result in decrease in antibody titers, both inhibition of antibody secreting cells in human PBMCs and decreased antibody titers in mice. We also showed that we could use polymeric nanoparticles containing rapamycin to basically allow for vector administration. And in this graph, which is kind of busy, you just have to follow the red line, showing that basically you could give twice the vector to animals with these rapamycin nanoparticles, and you can avoid antibody formation. This work was also published. I think I can go. And then this work was done at Geneton in collaboration with Otto Merten and the team, showing that, for example, one way to do plasmapheresis is to create columns that remove selectively anti-AAV antibodies, and the result is that basically you could do plasmapheresis not without depleting the total pool of IgG, and then just eliminating the anti-AAV antibodies. So I'm going to close in the last 27 seconds of this talk by, first of all, saying that we don't use rats that are 20 kilograms in the lab. This is just me putting the image too big. But when you look at the translation from mice to humans, I think there's a few considerations to be done. The first one is that both mice and humans, as I show you in my talk so far, they are very useful for proof of concept studies and to evaluate transgene immune responses and perhaps antibody responses to AAV. The larger, so a rat, a dog, a monkey, animals that are a better predictor of the therapeutic profile of at least AAV vectors, and again, they can really help you in are the immune responses to transgene and the antibody responses to AAV. And then there's a number of factors that you're basically stuck to test in clinical trials, and of course the immunogenicity of our vector, especially when it comes to cell-mediated immunity, is something that we learned that is mostly, we can only see the end result of that in humans. There's environmental factors which animals are really not exposed, and disease-specific features that, of course, sometimes animal models do not predict. So the idea is that, of course, as we all know, we go to the clinical trials and we have to go back to the bench and try to address potential issues, again, in the preclinical. Acknowledgements, all the team involved in immune studies and the people from Selecta that help us with the nanoparticles and fund institutions. The work done towards the trial of Crigler-Najjar was funded by Geneton, the FM Teleton, and by a European project, a consortium called CureCN. And the number of people involved is enormous, but I would like to acknowledge the Geneton, the people from AMC, ICGEB, Genosafe, the patient associations, and also, last but not least, the clinical investigators involved in the study, and the support also from the patient organizations, and finally the patients who really supported this project from the very beginning, and of course you for your attention.
Thank you very much, Federico, for the very nice presentation. Are there questions in the audience?
Yes, Jimmy. Hi. Whether the rat would be more So specifically, this is a very good question. The question is...
Could you repeat the question?
Yes, I was going to repeat. Yeah, of course. The question is whether we look into whether the rat is a good predictor of gene transfer in other organs, not only the liver, but for example, the muscle and so on. I don't have direct experience about the extent of transduction of other tissues. However, I know that there's a lot of work towards the development of rat models of different diseases such as Duchenne muscular dystrophy and Pompe disease and so on. And usually what happens is in some cases the rat will have disease features a little closer to the human based on, for example, severity.
One question at the back.
Say it again. Sorry. Yeah, the rat is a natural carrier. Or what makes them survive? That's a good question. I know that you can trigger toxicity in the rat by essentially, I forgot the name of the compound, by injecting a compound that would displace unconjugated bilirubin from binding albumin, because usually that's how it circulates, and so that, and then the animal will develop neurotoxicity. Whether this is, I mean, I think is, that would tell you that potentially they can develop neurotoxicity like the mouse. Maybe there's a modifier genes that makes the rat more resistant. I actually don't know. Gloria.
I think we should finish now.
Okay, the question was the lowest limit of inclusion as far as age is 10 years. Whether we think that at 10 the liver is fully formed, is fully grown to the size of an adult. The answer is no, because in fact there's quite a linear relationship between the size of the liver and the size of the body. I mean, up to a certain point, of course. However, I think there's a couple of points. First of all, is almost the size of an adult, and then during the teenage is when all the organs reach approximately the size of an adult. And then I think it's related to also the actual development of the liver, where you have a phase of cell proliferation followed by the polyploidization, and then the cells grow in size. And so the idea is that 10 will be a moment when you are past this phase, and then you will be more stable as far as gene transfer. Of course, we're trying to address that also in animal models and trying to do detailed studies. I show you a snapshot, but it's just the tip of the iceberg of the work that we're doing. And then ultimately, we'll have to test it in the clinic, but knowing that 10 is probably a very safe starting point when it comes to the risk of vector genome dilution over time.
Okay, thank you very much.
Thank you.
I think we need to move on to the next speaker. Thank you very much for the nice studies, particularly with the relevance of the studies in newborn animals. Those are very relevant. Thank you very much.